So today we're going to follow the journey of one drug compound as it makes its way through the process of lead development and FDA approval. So we've all heard conspiracy theories and false narratives that suggest that large pharmaceutical companies are hiding cures for diseases such as cancer so that they can continue to make money on existing treatments that cannot actually cure the disease. However, many people fail to realize the difficulty of developing drug compounds that are used to treat human disease and the myriad of ways that candidate drug compounds with high potential can fall out of the pipeline. So it's estimated that a pharmaceutical company will need to investigate over 10,000 lead compounds to develop just one or two drugs that might make it to market and be useful for treating human disease. And you can see that many of these candidates fall out during this early research phase or even in phase 1 clinical trials, largely due to unforeseen complications with side effects, low bioavailability, or failure to even reach the target site. And each drug that does reach the market does so with a very high price tag on the order of $2 billion or more. And it takes decades on average to get there, with some taking much longer. Why does it take so long? So today we're going to talk about the journey of one drug candidate for the treatment of hepatitis C. We'll first discuss the biochemistry of hepatitis C and different treatment strategies for the disease. Then we'll take a look at the chemical structure, the structure-activity relationships, and clinical development of this unique molecule. That this one contains a boron atom, which is really rare, as boron-containing leads are generally too toxic to move forward with. However, boron is unique in that it can often create stable yet reversible covalent bonds, unlike many carbon drugs. So the company that invested in this lead compound development was GlaxoSmithKline, or GSK, and they've developed many notable pharmaceuticals on the market, including the antibiotic amoxicillin, the antiretroviral drug zidovudine, used to treat HIV, and the antidepressant compound bupropion. However, pharmaceutical companies are also never free from corruption or greed, and GSK is no different. GSK settled one of the largest healthcare fraud allegations in a $3 billion settlement for the unlawful promotion of certain prescription drugs and its failure to report certain safety data. This is why the drug industry as a whole needs to have careful oversight by the FDA. So both the good and the bad can come from the pharmaceutical industry, and it's not free from greed or needing to show a profit from investments. Okay, so let's jump into today's topic, hepatitis C. Hepatitis C is a severe disease leading to liver deterioration and serious complications. It's caused by the hepatitis C virus, which we will call HCV. And so HCV chronically infects over 170 million people, or about 2% of the world's population. And the dilemma is that the disease incidence and the drug market are inversely related. The key Western Pacific, Southeast Asian, and African regions have the highest prevalence with 130 million infections and almost no access to HCV drugs. The United States and Europe, on the other hand, have around 15 million infections, but are the key target market for drugs. Worldwide, treatment is complicated because HCV occurs in 6 different genotypes or variants, which are also distributed unequally geographically. You can see high-income countries have primarily genotype 1, which is only about 10% of the disease burden, whereas in low-income countries, variants 3 and 4 become much more prominent. These different genotypes influence drug sensitivity and resistance. Thus, Seeking treatments favors drugs that primarily target variant 1 over the other genotypes that are more prominent within the world. So the Nobel Prize in Medicine in 2020 was awarded to Harvey Alter, Michael Houghton, and Charles Rice for the discovery of the hepatitis C virus. HCV spreads via blood-to-blood contact, so injection drug use, poorly sterilized medical equipment, and other blood-to-blood contact pathways lead to disease acquisition. In contrast to HIV or hepatitis B, HCV is not transmitted sexually. An infected individual can be infected for decades without obvious symptoms, while increasing risks for liver failure and cancer. But the good news is that unlike infections with HIV and hepatitis B, HCV can be cured. However, while there are approved vaccines for hepatitis B to prevent it, only few are still in development for hepatitis C. Early physiological studies were recognized with the Nobel Prize in 2020, and the last 2 decades truly saw a revolution of HCV drug discovery. As HCV is a viral disease, there are multiple potential inhibition points in the virus life cycle, including entry into the host cell, protein synthesis steps, and packaging and release. However, if we contrast HCV drug development with HIV, it becomes evident that there was quite a slow start. While the first HIV drug was released just 3 years after the virus was identified, it took 24 years from the discovery of HCV to the approval of the first treatment. There are many reasons for this. First, low perceived market value, low pressure from patient associations, but also a poor understanding of the nature of HCV as a disease. There were far fewer basic research studies. Advancements in structural biology, pharmacology, and also clinical trial design ultimately led to the approval of various therapies. Most are combinations based on interferon proteins, which work by increasing immune defense, administered by subcutaneous injection, which work by increasing immune defense and are administered by subcutaneous injection, and ribavirin, which is an oral, broad-spectrum antiviral that is used against various viral fevers. This one-size-fits-all combo, launched by Roche Pharmaceuticals, was priced at around $40,000 per treatment and had a decent efficacy of about 55%. But true improvements came only with the development of combination stacks with additional direct-acting antivirals, which are more targeted and robust against resistance. These also came with a half-treatment duration as the third evolution. Oral combinations were also developed. This bottom treatment strategy was bought by Gilead from Farm Asset and marketed as Sovaldi and Harvoni. The market upsurged in 2014 with the release of these drugs and peaked for sales of $23.1 billion in 2015. They have continued to have a steady market presence ever since this launch. The cost of each treatment is still around $40,000, but delivered an even higher efficacy rate of 89%. So why, if there are already blockbuster drug treatments out there for HCV, should more development go into new agents to treat the disease? All of the remaining untreated tens of millions of HCV-infected individuals can't afford outrageous prices of up to $50,000 per treatment. Neither can the World Health Organization, or WHO, who aspires to treat 80% of diagnosed populations by 2030. The development of new direct-acting antivirals thus may serve to increase competition and help to lower treatment costs. This may then enable more equitable access to medicines. Also, many of the drugs developed were not equally effective against the 6 different genotypes of the virus. So there's still a lot of work to be done. So various potential targets in the viral life cycle can be targets for antiviral development. GSK chose to focus on the retroviral RNA polymerase as the primary target for the drug discovery, and in 2014 they published initial studies of inhibitors to the NS5B polymerase. You can think of this as the viral RNA printer. This is the same mechanism of action of Gilead's Sovaldi. The structure of this enzyme was first discovered in 1999 and has a hand motif with the section noted as the palm, the fingers, and the thumb. Inhibitors, as you've learned, can bind directly to the catalytic site of an enzyme or to an allosteric site that alter its activity. In the case of NS5B, there are 4 well-established allosteric binding sites, and the POM2 site is particularly interesting. It's interesting because it's closest to the active site, and there are many amino acids that are highly conserved in this region across the HCV genotypes, so a potential inhibitor could combat all variants effectively. So you can see the active site of the enzyme in yellow here, and the allosteric inhibitor binding site noted by the number 3. Some of the conserved amino acids are also indicated by their 1-letter code and their placement in the peptide chain. For example, this is arginine, position 386 in the protein's primary sequence. GSK identified this inhibitor number 3, which nicely docks into the POM site. This impacts NS5B enzyme in 2 ways. First, the binding of compound 3 stabilizes the closed state of the enzyme, which is much less active than the open state. And 2, the head group of the inhibitor also interacts with the catalytic magnesium cofactors and the active site, thereby disrupting the placement of incoming nucleotides. This makes polymerase initiation and propagation more challenging. So what do these inhibitors look like? They started with a benzofurane core with the cyclopropyl group that was initially described by another group as having antiviral properties. When they substituted an alcohol group at the position indicated in yellow, they saw strong activity against the wild-type genotype 1 HCV, as well as a common mutation of the virus that occurs at the 316 position. The activity was assessed looking at cell-based replication systems. As well, the activity was assessed by looking at cell-based replication mechanisms, as well as data from an in vitro biochemical assay of the enzyme. As mentioned previously, the nonpolar region of the molecule was bound in the gray palm side, while the polar head group sits in the active site. By screening different head groups, the GSK group found that by appending a boronic acid, activity was pushed into single-digit nanomolar range, even at the common mutation position 316N. Remember that the lower the concentration indicates the stronger the inhibition. Para-substituted aryl boron derivatives were also more effective than alcohol functional groups, especially for the wild-type 1A variety. However, the meta-substituted aryl boronic acid derivative showed reduced activity, highlighting the need for proper orientation of the head group within the active site. What about the need for the boron? When substituting boron for other polar head groups, activity in the biochemical assay remained high. However, the inhibitors had significantly lower potency in the replicon assay than the boron-containing derivatives. This could be due to lower cell permeability of the charged polar group in the phosphate derivative. Boronic acid has much weaker activity and did not suffer the same delivery challenges. However, drug activity is only one aspect of a good drug candidate. As you've been learning, the pharmacokinetic profile of a drug is also very important. GSK found that the boronic derivatives suffered from low bioavailability in the rat model and were rapidly cleared and excreted. This challenge was significantly improved by adding a fluorine to the phenyl ring, increasing bioavailability 5-fold, while even further increasing the potency, especially against the 316N variant. They tried to further increase activity by placing the boron in a ring structure. This resulted in better performance in the biochemical assay, but a weaker performance in the replicon cell-based assay. So GSK decided to move forward with compound 3 in their studies. The team performed crystallization experiments with the enzyme to help reveal the binding modes. There are various interactions that contribute to the function of the inhibitor. These include the interaction of the fluorine in a hydrophilic cleft within the molecule, as well as polar interactions with the amide functional group. They also found that the benzofuran structure is anchored with an arginine amino acid. This arginine is conserved across all 6 genotypic variations of the HCV, making the team hopeful for broad spectrum activity against all HCV variants. The boronic acid did not form any notable complexes with the enzyme. However, it did form a hydrogen bond with a bound or structured water molecule in the enzyme active site. So this looks like a real promising lead, right? Unfortunately, no. In human trials, they noted that the drug had a very short half-life of less than 5 hours, requiring the delivery of a much higher effective dose. In addition, they found that the metabolism of the drug resulted in a major metabolite with a very long half-life. Unfortunately, this metabolite was also observed in phase 2 clinical studies of another drug compound and was associated with high liver toxicity. Although there was no toxicity observed with this compound, the team wanted to limit the potential toxicity of this compound and limit any adverse reactions. So they tried to modify the compound further. The key step was to prevent the oxidation of the benzylic carbon. So the team wanted to maintain the same biological activity of the inhibitor and not start over from scratch. So they hypothesized that they could make 3 potential modifications to their existing molecule that might prevent this oxidation reaction and the formation of the unwanted metabolites. The first 2 consisted of cyclization of either the phenyl ring or in the direction of the sulfonamide, while the third route eliminated the carbon directly from the molecule altogether, shortening the molecule by 1 CH2 group. However, this would risk disrupting the positioning of the polar head group in the active site of the enzyme. They also considered just adding a methyl group to the position to shield the potential for oxidation. However, this reduced the activity by 50-fold. So, how did the other 3 approaches work out? Cyclization onto the aryl ring reduced inhibition significantly, especially for the 316N variant. So, no good. Within approach B, the team replaced the sulfonamide with carbonyl-containing groups because it appeared as though only one of the oxygens made meaningful contact with the polymerase enzyme. The results were mixed. For some analogs, the wild-type inhibition decreased significantly. You can see the numbers go up significantly here. For some analogs, the wild-type inhibition decreased significantly. However, compound 25 actually retained very good potency. So, compound 25 was quite encouraging. And approach C yielded better adjustments. Here, the para-substituted benzoic acid continued to perform better. Here you can also see the dramatic impact of electron withdrawing groups on activity, particularly for the two variants. And the chloro compound 47 was even more potent, causing the team to shift away from their use of fluorine. The final enhancement was to close the ring structure, which enhanced chemical and metabolic stability, and the shift did not lower activity too much. Looking at the crystal structure of the new lead compound revealed a network of interactions mediated by highly ordered water molecules, shown as the 3 red balls, which the authors did not observe with the other leads. The final test was to see if this compound resulted in improved pharmacokinetic profile. Within a rat model, they found that compound 33 and 49 had much lower in vivo clearance and were much more bioavailable. The researchers also compared cytochrome P450 activity across the compounds. Compound 33 had a micromolar biological inhibition of P450 enzymes, raising concern about potential drug interactions and risks. On the other hand, lead compound 49 was less active against P450 enzymes, posing significantly less risk. In human studies, they saw a much longer half-life of compound 49 and no oxidation to the potentially toxic metabolite that was seen with the previous compound 3. So the compound proceeded from phase 1 clinical trials into phase 2, where the GSK combined the treatment with an RNA-based treatment called RG101. However, after they observed 2 cases of serious jaundice, so patients turning yellow due to liver dysfunction, the FDA put a hold on RG101, and GSK actually also decided to not further develop their compound 49. Unfortunately, not the success story we are all hoping for. So, this gives you a real-world example of just how complicated and difficult it is to take a drug lead and turn it into an approved pharmacological agent. This also sets us up to discuss structure-activity relationships and their importance in drug development in the next few lectures.